Welcome to BioCode. In this video, we will show you how to use Prodigal gene prediction software. It is a software that is utilized through command line such as CMD on Windows, Terminal, and Bash on Linux and macOS. So to get started, what you should really know three things basically about Prodigal. Prodigal is a gene prediction software that is utilized for metagenomes and prokaryotes. It cannot work on eukaryotic genomes. It can work with drafted genomes or those genomes that have been already assembled into a proper genome. But to get a better results out of your genome, to predict better genes, number of genes from your genome, you should utilize a proper and finished or assembled genome rather than utilizing a draft genome. To get started, I will be providing you these notes that are basically handwritten to or curated notes that are to provide you a basic concept about how Prodigal works basically. Prodigal basically predicts coding genes out of your genome. Whatever genome that you provide, it will predict protein coding genes, and it will provide those results in a GFF format. If you don't know what that is, you can watch the file format videos, GenBank format or sequence table format. But to make sure that these formats are make sure that you select a particular format to be output for that particular results. Otherwise, by default, it will predict or it will output those predictions in a GenBank format. It can handle those genomes that are drafted, as said earlier. It can handle metagenomes, it can handle finished genomes or assembled genomes, but it is recommended to utilize finished genomes and finished metagenomes. Metagenomes are basically those genomes that have various genomic features of various species within a single assembly. So it is quite efficient on those genomes as well. It is very fast as compared to other gene prediction softwares. Prodigal can predict E. coli genome genes within E. coli genome within 10 seconds. That basically, if you run it on a MacBook Pro, depending upon the processors or RAM you have on your computer, so it is quite efficient and it is quite quick to provide you the results basically. If you compare the results with other softwares or other gene prediction tools, it does not really. The most benefit about the best benefit or the most advantageous fact about gene prediction and Prodigal gene prediction software is that fact that it does not need training material with it. You can provide training material, but I will tell you why we need training material. But it basically runs without any, it is so unsupervised. I will tell you what does that mean. But it does not really need training material. For example, you don't really have to provide any material, any outsourcing or third party material or third genome material to your Prodigal gene software prediction tool so that it can predict according to your input. Rather, all you really have to provide is the genome file and it will predict on its own. You don't really have to provide any training data. Any other properties of any particle genomes are not provided. It will predict the genes based on the entire properties of the genome that you provided, or the RBS, which is ribosomal binding site, motif usage, and it will predict the basic on the start codons, ORF, or coding statistics of the prokaryotic genomes. So unsupervised mean in the machine learning term, unsupervised mean that you don't really have to provide a particular data set for the training of the software. It works without any data set. It is a machine learning based algorithm, by the way, that is Prodigal is based upon. So you don't, if it needed a training material, then it would have been a supervised algorithm rather than unsupervised in this case. But there is another aspect of Prodigal in which you have to provide a supervise in which you have to provide a particular data set for the training of Prodigal. That data set can be the known genes of a particular related genome. In that case, you have to provide a data set, but it is only applicable in that scenario in which you have a shorter genomes. For example, it is shorter than 300 kb, 500 kb. In that scenario, you can provide in that scenario you can provide, then you should provide a data set that is related to that particular genome and it has the genes already predicted, so that you can the tool will compare and learn from that data set to predict from the genome of interest you already have, that you provided as input. We will talk about that in the next section of this video. But it can handle gaps or partial genes as well. It can basically ignore ends that are repeated mask regions, and it can ignore those regions within the genome, and it will not use them to predict the genes out of your genome. And it can identify translation sites, such as those sites that actually are translation sites, for example ATG, or basically they start from they are transcription sites, but they also become a translation sites as well. So the output that is provided in the Prodigal is basically conditional scores, confidence score, RBS motif that I told you about, the ribosomal binding site, and other statistics are also provided. The output can be protein translations of the entire gene set that has been predicted through this tool, and the genes as well in another particular file. We will discuss that in the next section of this video. So let's move on and let's open this second note. And as you can see, there are three particular modes within Prodigal. Prodigal works normal mode, anonymous mode, and training mode. Normal mode, in which all you really have to do is provide the genome sequence, and it will study it, learn its properties, and then it will predict based on these properties. Otherwise, there is a second scenario in which you are working as anonymous mode in the Prodigal software. It will apply pre-calculated training files that, as I said, it can have some of its own files, but you can provide your own files. Anonymous mode should be applied when you are working with genomes that are smaller than 300 kb or 500 kb. Bacterial genomes can be smaller than that, or prokaryotic genomes can be smaller than that, keeping in mind that Prodigal only works with prokaryotes. And there is a third mode, and that is training mode, which works like normal mode, but requires a training file, but saves a training file for the future use. For example, you have determined a gene prediction of a particular genome XYZ, and you used a normal mode, but you should have used a training mode. In that scenario, what would have happened is that the mode would have actually predicted the genes from your particular genome of interest, but it would not have required a particular gene set, but it would have saved those statistics for the later use. So in case next time you would like to use anonymous mode, you can use the prediction data set that you have already created. So that is how these three scenarios work. And let's move on to the third note. It really depends upon what you really want to do with your data set, and only that scenario can be utilized to outsource what mode should be utilized. So for example, as I said, normal mode should be utilized when you have sufficient data, and Prodigal depends upon, for example, 100 kb plus. If you have a 100 kb plus data set of 100 kb means kilobyte, kilobases. So in that scenario, if you have a prokaryotic genome that is bigger than 100 kb, it will be good for 3' predictions. If you have a 500 kb plus genome, then it will be good for 5' predictions. Otherwise, you should utilize the anonymous mode, because that mode can require the training scenarios, and using those training data sets, it will predict a gene set to be a better efficient set. And if you are working with metagenomes, anonymous mode should be used, because metagenomic data set can vary from metagenome to metagenome. So that is why you should utilize anonymous mode. So let's see our fourth node, which is normal mode should be only utilized when you have finished genomes. For example, as I said earlier, it can work with draft genomes that have not been finished yet, or it can work with viruses as well, but it should be only utilized for the finished genomes. Anonymous mode can be utilized with metagenomes, low quality draft genomes that have not been completed. Small viruses are quite smaller when we compare them to other prokaryotes, and small plasmids can be utilized as well for the gene prediction, but these methods should only be utilized for anonymous, not normal mode. Training mode, which is the third option, which basically works like normal mode, but the output file is a training file as well that you can save for your later analysis later, and you can use this file in the anonymous mode as well. So that is how you utilize these modes. It really depends upon how and what sort of properties within your dataset are present. So for example, suppose you have already installed Prodigal. We will discuss about the installation, but let's get these notes done. You can find these notes related information within the transcription. By the way, all the links that are required can be found there as well. Prodigal is a command line tool, as I said earlier, so it requires basic parameters that should be defined in the command. For example, P or dash mode define what sort of mode you would like to use. By default, it is normal mode, and as I said earlier, as I discussed earlier, normal mode requires just the genome or any number of sequences that can be present within a FASTA file, or they can be various files as well, but they should be a genomic data, not gene itself. The training data requires the training and the input of the file that you would like to be predicted, and the anonymous mode requires the training data set and the ideally metagenomic data. And there are other parameters that have been deleted and become unknown and normal, so you don't really need them. And these are the parameters that are utilized. For example, input dash I or dash input file requires the input file that, for example, the genome that you are going to predict into genes, and the output file, what sort of output file you want, where you want the proteins, and that is utilized through dash A. Dash D outputs the nucleotide files. Dash S outputs the start file, and this dash W is utilized for the output of summary statistics, and the dash F requires if you want the output to be GenBank, GFF, Screen, or simple coordinate output, as we discussed over here, that GFF, GenBank, or Sequence table can be utilized. So this is how you can utilize to output your results in a particular format. Dash Q is utilized when you have quite a bigger genome, and so you don't really want that the results to be the processing to be displayed on your terminal, so you can just utilize dash Q and get away with the results when they are done. So this is the basic command that is utilized to prediction into a normal mode. We are only going to discuss the normal mode in this video. We are not going to discuss the anonymous or the training mode as well. So I already have installed it on my computer, and it is a Linux server, a Ubuntu server, by the way. You can install it on your Windows, Mac OS, or Linux computer as well. You don't really require a server. I'm just going to utilize it because of the fact that I already have installed on there. This is the link that you can find in the transcription that you can utilize to download this Prodigal software, which is available through installing Prodigal page. That will open up another page. From there you can install it on Mac OS, generic, or Windows. To install it on a Linux computer, it is quite easy to actually do that. Let me show you. If you do sudo apt, and to sudo apt, all you really have to do is call in the Prodigal name, and it will install. There we go. I already have installed, so it won't really be reinstalled. So in that case, you don't really need to reinstall if you already have it. So I already have it. I already have done some analysis, but I'm going to do it again for this video. So let's open it up, and from there we can move on. Let me remove these GPK files. Okay, so this is a particular genome that is that is a genome of Lactobacillus fermentum, which we retrieved from the NCBI genome FTP. You can do that as well, but in most cases when you are doing gene prediction, you don't really download a particular genome from a particular dataset. You have your own genome because you have just sequenced it. In some cases you can download it, but that this is basically how you do gene prediction for the particular genome. I'm going to utilize this particular genome. Let's head over it and let's show you if it is a particular genome. So this is, as you can see, this is the complete sequence of Lactobacillus. This is the accession, you can utilize it to download this particular genome. This is the complete sequence, and let's count the exact number of letters and lines within this file. As you can see, over 29,984 lines within this, and now over 21,200 KB of sequence of letters are present within this sequence. So we are going to utilize this sequence for the prediction of genes. Let's call in our command, that is basically, as I said earlier, this is the basic command that is utilized, but we are going to make changes to it later on. We are going to output results in a different way as well. I'm sorry. Okay, so let's get started. Let's ls over it. This is our sequence that we are going to utilize. Let's call in the Prodigal software, Prodigal tool. Then we have to provide the input file, which is going to be the sequence dot fasta, and our output file should be, if we want the output to be GenBank format, which is by default, so we are going to utilize the GenBank format. Let's output genes.gbk. Then we also want the protein translations of these particular genes that have been predicted, because in most cases you also require the genes as well, and proteins as well. So output, and let's make it output proteins and FFA, which is basically the FASTA format of the proteins. So all you really have to do is just press enter, and it will start analyzing your sequence. It will take some time, and as I said it earlier, it is quite efficient in predicting your particular genome, particular genes out of your genome. So let's ls over files. These two files have been created, so let's visualize them. Let's cat and output genes.gbk. So these are all the genes that have been predicted through this particular genome, through this particular software, and let's count how many of them are there. Grep cds, and we can do count. Our file was output genes.gbk. So a total of 2094 CDS have been predicted within this particular genome, and let's do proteins as well. Let's first visualize our protein file. Cat output proteins dot far, and as you can see, these are the proteins that have been translated from the genes that we predicted using this tool. So let's find the static. Basically means the stop codon. It tells you that this is where the protein translations was terminated. So this is the basic accession that was of the genome, and the numbering is, as you can see, this is the numbering 91, 92, 93, 94 in a linear way. If you go up, this will go in the other orientation. So the total of 2094 proteins have been translated from this genome. Then the start position is mentioned of the protein, and the ending position is also mentioned, and this is the strand, if it is present on the negative strand rather than negative strand, and the first and the first translation